That I would now like to introduce you to Alice McHardy, who is our first speaker today. So Alice currently is at the Heinrich Heine University in Düsseldorf, and formerly was at the Max Planck Institute for Informatics. Is that right? Currently holds the chair in algorithmic bioinformatics. So her interests are currently in microbial evolution, and previously is it previous or still currently?
It's still going on, yeah.
Still going on, taxon binning, and in genotype to phenotype interactions, and also biomass.
Yeah, that's an application of.
That's an application. I guess we're going to hear something about some of those things.
Yes.
So thank you very much, Alice.
Thank you for the introduction, and I would like to thank the organizers for inviting me to this great workshop. I really also enjoyed all the talks that I've been attending so far, so thank you. So today I'll be mostly speaking about the inference of genotype-phenotype relationships, which I think is, from a practical perspective, the thing that you really want to get out, or many biologists and medical people want to get out of the data when they start to sequence this. So it's a very important problem, which genes are related to which proteins, to which protein families, to which biological processes, and so one should think about how we can learn these things from these data sets. You probably know these questions. People ask them to motivate their slides. So who's there? You want to know about taxonomic communities, and there's ways to analyze it using marker genes, amplification of informative marker genes, or you can learn this of shotgun metagenome data. And then also what is the community doing, what are the different community members doing, and then we have all these analysis techniques, these meta techniques, metagenomics, metaproteomics, metatranscriptomics, and the reconstruction of draft genomes is somewhat, it's usually performed using assembly and binning techniques, and I'm one of the fans of doing assembly of metagenomes. I realize it's not possible for many samples because they're so complex, but the value is just much larger, what you can do with it and what you can get out of it. And if you have the draft genomes, because if you don't have that, you can do this bag of gene analysis basically for the entire metagenome and get some indication of what functionalities are there. But if you have the individual genomes, then you can really think about how the individual community members, how they contribute, and what the individual members have, and make some also inferences maybe about the evolutionary rates, what we've also heard some talks about. So you probably know that it's for this community, this is kind of obvious. After the sequencing and the assembly of the reads, of generation of the reads, we have different computational steps, such as assembly. May or may not do this. If you don't do this, you go straight here to the analysis. Taxonomic assignment, which means assigning taxonomic identifiers to either individual reads or shotgun fragments that you've obtained from assembly, and to annotate them, so to assign functions and pathways. And maybe at some point somebody will come up with a really cool Bayesian method that integrates over all of these possibilities and then does it all at once, who knows, and we deal with all the noise. So we've been looking at or developing methods for the taxonomic binning of shotgun metagenomes, and I will not just mention this very briefly, because in the end I want to talk about an initiative, a community initiative for assessing computational tools, but then go into this problem that we've also been looking at, which is inferring genotype-phenotype relationships in terms of protein families that are relevant for the establishment of a particular phenotype. And in particular, we've been focusing on the phenotype of plant biomass degradation by microbes. This is a phenotype. We chose this because we have experimental collaborators who work on this, and we had also some metagenomes available from plant biomass degrading communities, so it seemed an obvious choice to look at. And so you want to infer the genotype-phenotype relationships, and if you do, you can also build you can also start to assign phenotypes to genomes, so using some classification functions. And the work that we've done here has been published last year, and this is one part I'm going to describe. I will not talk about microbial evolution yet, because this is sort of this emerging field in the field, or linking different fields. I think it's exciting, but I don't have anything so far to show. Yeah, taxonomic binning, again as a slide, you want to assign fragments to different taxa, and then if you have shotgun data, then the set of fragments corresponds to your bin. So you can also say it's to deconvolute your data set into bins. And our method that we had back then is called phylopaetia, and the idea was that it uses k-mers and looks at k-mer profiles, and these are taxonomically informative. So this was known back from the works, I think Samuel Carlin did a lot of work on that, and found that short oligomer distributions are species-specific and rather homogeneous across an individual genome, and also in some cases for higher-ranking taxa. And you can use this information to learn a classifier for different taxa, and then because it's a property that's global, you can take any piece of the genome, train your classifier, assign any other piece of the genome, and you do not need and closely related reference genomes. That's one of the benefits. Nowadays, we of course have also different approaches. We'll hear more about that, like using variations in sequence coverage. Alex Gerber mentioned some of that yesterday also. Or you can also use sequence similarity. So this gives you using local sequence similarity for taxonomic classification, like Daniel told us about. And this has an advantage also in some scenarios, so that this is a stronger signal than k-mer usage, in my opinion. So it's more informative across large evolutionary distances. So it depends on what you want to do with your data and what you want to get from it. So why is microbial plant biomass degradation an interesting phenotype? It is because it can be used for the generation of biofuels. So it's a renewable energy resource. Sun photosynthesis is going on, and grasses are being, can be regrown easily, and they can be broken down into lower molecular carbohydrates, and from these you can generate ethanol, and ethanol can serve as a biofuel. And you want to use lignocellulosic biomass, so not sugar or corn, for instance, because these are not food sources, whereas corn or starch you can also use to feed the world. So you want to make use of things that people don't want to eat. And therefore there's a lot of interest in understanding how to degrade this plant biomass in an efficient way into individual carbohydrates to go from there and to generate, for instance, biofuels. In microbes, they have figured out how to do this efficiently. And so it's very interesting to find the genes that are relevant and implicated in microbial plant biomass degradation with the prospect of finding things that can help to improve industrial processes. And we're currently looking also at other phenotypes that are relevant for other plant-related phenotypes, mostly because we are at Heinrich Heine University. We're sitting in a cluster of plant biologists who are very good at looking at this topic. And this work that we've published, it's been, well, we haven't been given any award, but we've been invited to apply. And this was an award by a company that gives awards for research paper that relate to alternative energy generation. So I thought it's really nice that you get recognition from such an applied perspective. Okay, so why is the degradation of lignocellulose in plant biomass difficult? And you see here the structure of plant biomass from a paper from Eddie Rubin, actually, that he published in 2008. And so it has different components. It has three major components. One is there's these cellulose fibers, so crystalline cellulose. These are long chains of just glucose molecules. It's very homogeneous, and it builds like a crystal structure. And then you have so-called hemicelluloses. This is more diverse and includes different kinds of sugars, and also more branching structure, not so nicely ordered. And then there's a third component, which is called lignine, and it includes different kinds of aromatic compounds that then build this kind of network, and this cross-links also between the hemicellulose and the celluloses. And then the whole thing is organized on different levels, so you have microfibrils of these cellulose fibers, and then this is all part of macrofibrils and part of the plant cell wall. So that's the structure. And the large-scale degradation in the industrial setting, it usually takes a bit strong conditions. So you need acids and a bit higher temperature to break down this material, to make it accessible for the enzymes to attack it and degrade it in individual sugars. And high temperature means high energy, and that is wasteful and costs money. So you want to be able to do it without using higher temperature or other things. And so the search is for new catalysts that can efficiently break down these recalcitrant materials. So just some overview of the structure of these different polysaccharides. So you have cellulose, I said, is a homopolymer, so just glucose molecules, and then you have the different kinds of hemicelluloses. Each of these symbols here denotes a different kind of sugar, so you can see that they're very heterogeneous. And then there's also, in smaller amounts, something called pectin, which is also a heterogeneous polysaccharide. And microorganisms that can degrade linear cellulose, they therefore need a cocktail of different enzymes that can attack these different substrates and break them down until you end up with the individual sugar molecules that can then be converted and go into the metabolism into other things. So microbes, there's two well-characterized paradigms how they are able to do this. There's one is called the free enzyme paradigm, and as the name says, it involves a couple of enzymes that the microorganism secrete to the exterior of the cell, so where the cellulose is located. And you see here now just from the cellulose substrate, so the cellulose fibers, then you can see that this is regions where it's crystalline, it's very nicely packed, and then here it's amorphous. And you have the different enzymes that attack different parts of the structure. So you have the exogluconases, they attack from the ends, endogluconases, they attack in the middle, and then you have beta-glucosidases, which break down then if you just have sugar dimers left. And you can see the endogluconases, they prefer to attack in this amorphous region. And similar strategies is employed in the so-called cellulosome, with bacteria using these cellulosomes. And there are similar enzymes involved, but there's also something called the cellulosome itself consists of two proteins, coenzymes and dockerines, and these basically serve as a docking station for all the different enzymes. So the enzymes, they have a so-called carbohydrate binding module that's attached to them, and this can attach to the dockerine module of the cellulosome. So these are like adapters, and the enzymes therefore get concentrated at high concentrations here locally, and then they're more efficient at degrading this material. But it's not known for some of the bacteria how they do this. So other bacteria, they don't have these key enzymes families like GH5, GH6, or the carbohydrate binding modules. So we thought we could take data and try to see if we can associate certain protein families with this phenotype and learn from the data. There's also some new emerging paradigms. So people are trying to find out how these bacteria, where it's unknown how they do things, how they do it. And one of the indications is that there might be some system that's called SAS-like, similar to a starch degradation system. Starch is also a polysaccharide made out of sugar units, but a slightly different structure. And there's enzymes on the outside of the cell that degrade it, and then it's transported into a periplasmatic space for bacteria that have two kinds of membranes, so these are gram-positives, and then there it's further degraded until it gets into the cell. And there's different sets of enzyme involved, and there's research into whether this might explain, this might be how some of these bacteria perform plant biomass degradation. Okay, so what we did was these two people here, Aaron Byman and Julia Turkina, they worked on training a classifier or developing a method that allowed us to, first of all, predict whether an organism is biomass degrading or not, given genome annotations, and then secondly, to identify the relevant protein families that allow to distinguish, or that distinguish plant biomass degraders from non-degraders. So what they did was, first of all, they went to the literature and they extensively searched for known degraders, so using information from databases, going through articles on plant biomass degraders, and they came up with a set of known degraders, and then they also came up with a set of probable non-degraders, which is much more difficult to find evidence that an organism cannot do something. So they were fairly, well, they didn't find any evidence that this organism could grow on these kind of substrates, and that was our set of non-degraders. And then they trained the linear support vector machines, or they annotated the genomes of these organisms with protein families, so PFAM domains and KC families, and the counts of each protein family in these genomes were the features. And then you can train a classifier to distinguish between genomes of degraders and non-degraders, and they used a linear support vector machine with an L1 regularization term, so you get a more sparse solution. And then, from the weight vector that you learn in this primal representation, you can just select all the features that have been assigned positive weights as potential candidates. And then you have this issue that if you train a multivariate classifier, it may just select one of many correlated features. So, but you can improve by training a whole set of classifiers using cross-validation, using different parts of the data, and then you can select features that occur in multiple of them, so as basically consensus features. And this is what they did. And so these consensus features of these classifiers represented our candidates' features that jointly allowed to distinguish between degraders and non-degraders, and potentially, if there's a link between classification ability and functional relevance, could also be protein families that are implicated in this process. And of course, these classifiers could also predict the capability to degrade plant biomass. And we used to train these parameters standard nested cross-validation experiments, and had then good performance in these experiments, so high accuracies of over 90%. So here you see for the Casey families, which is the first case what we tried. This is a database of so-called carbohydrate active enzymes, and it's a bit hard to read. I'm going to explain to you what there is to see. Up here you see the protein families that were selected by the classifiers as relevant features, so that were attributed positive weights in the majority of them. And these are the genomes of cellulose degraders, and these are genomes of non-degraders. And you can visually see this heat map shows you the occurrences of them across the degraders, non-degraders. So you see, as one would expect, they're more abundant in the degrading species. And here are protein families classified based on their functional context. So CASI database includes protein families relating to all the degradation of all kinds of polysaccharides. And so the yellow ones are cellulases, and you can see some of those I mentioned before in the introductory slides. So for instance, you have GH5 here, which, so this one is found, for instance. And also some carbohydrate binding modules, so you need to attach to carbohydrate substrates, and then oligosaccharide degrading enzymes are the green ones. So that seemed sensible. And we also applied the same thing for domains we annotated from the PFAN database. And you see cellulases, hemicellulases, oligosaccharide degrading enzymes again, and carbohydrate binding modules. And then now what the interesting thing is, there's also functionally uncharacterized domains in here. So these now could be candidates or are candidates. Potentially they're relevant for this degradation pair or for the degradation of linear cellulose, and should be considered with further experiments. So one I personally like is a pasta domain. I'm not sure why it's relevant, but it sounds nice. We have experimental. We're working with an experimental lab that characterizes, does biochemical characterizations of such enzymes, and we want to test them. So the second thing is that we predicted plant biomass degradation, and we used a data set that Alex Gerber put together, who's sitting over there. I think you were binning it, right? So this is your work. These are Alex's bins. And so basically it's a shotgun metagenome of a microbial community that was isolated from, that was shown to degrade plant biomass and isolated from cow rumen. And these are bins, draft genomes of 15 different ones, and we applied our classifier to annotate these, or we annotated these draft genomes and then classified them whether they're plant biomass degrading or not. And so double plus means all classifiers assigned it as plant biomass degrading. 1 plus means it was, we used 5 classifiers, so it was 4 out of 5, and then you have the negative predictions or ambiguous predictions. And you can see, so the blue ones here, these are taxonomic bins where we could directly map to gene sequences that were, for which the proteins were shown to have cellulolytic activities in that same study. So that is one confirmation of the results. Then we have one bin here which was predicted, but which we had no confirmation for, except that it was also mentioned in the original study as being closely related to a known degrader, butyrovibrio fibrisolvents. And there are a couple of predictions which, so these two we don't know about, but they seem to be already, seems to make some sense with these results. Then a related topic is a student, Sebastian Konecny, has been working on. And as a first step, what we want to do is infer protein families that are functionally coupled. So in the study I just showed you, we looked at protein families that individually could be relevant for the plant biomass degradation phenotype, but there's more information in genomes, so you have also protein families tend to act together and in concert, and there's also signals of that in the genomic data, which is information that is also used in the whole NGRAS machinery, for instance. So if you use it and look at the fig farms that they have, these were created also considering information of how families are distributed across genomes, what are the neighbors of families. So they use genomic co-occurrence across genomes or genomic clustering in the genome to infer, to place protein families in a functional context. And we wanted to apply this principle of looking at protein family co-occurrence patterns across genomes and also including metagenome bins to infer sets of protein families that could be related and placed in a functional context. For instance, that occur in a joint pathway or that are part of one protein complex. And to infer this, we used a probabilistic method, which is called latent Dirichlet allocation. And I think about, well, more the statistical inclined people here, they know this technique. So it works. Originally, it was devised to find so-called topics from a collection of documents, and it uses co-occurrence signals across documents to find words that tend to co-occur and that are therefore probably coupled. And I will tell you about how it works approximately. And it's very similar to the question we are trying to address, namely to find sets of protein families that co-occur across some of the genomes. But they will not be in all genomes because not every organism has the same capabilities. So to give you an example of what the data looks like, this is an example of the fatty acid biosynthesis pathway. You see here some substrates going in and out, and you have enzymes that are performing different steps on these substrates. They're color-coded here. And below, you can see in the genomes of different organisms, genes that encode these respective functionalities. And you see that they're co-occurring across the different genomes. And there's also a clustering within the genomes. And organisms that won't have this pathways will tend not to have these enzymes. Of course, these separations aren't perfect, the data is somewhat noisy, and so on. Not every organism has the same set of genes, and sometimes a gene may also be involved in a different pathway, but there's a clear signal that you can see and that you can use. So how does LDA work? LDA originally was this was devised to infer topics from a collection of documents, and the idea is that each document basically can be described as a mixture of being generated from a collection of so-called topics. And you see examples of these topics here. So a topic is a probability distribution over a fixed vocabulary, so over a set of words. And in each topic you have different probabilities or high probabilities for different words. So this is a topic where gene, DNA, genetic has high probabilities. This is a topic where rather brain, neuron, and nerve has high probabilities. And now in this model that LDA assumes, the underlying process that generates the collection of documents, you have for each document, you first have a distribution over topics, so which topics are relevant for generating the content of this document. So for instance, for this example here, for this text, one would assume that this topic has high probability, and the yellow one and a bit the blue one. And then you select one of these topics, and then from the topics you draw a word. And this is, if you have a document of n words, then this is how you generate the document content. And so we want to apply the same principle for genomes and taxonomic bins. So our documents are the genomes, or the taxonomic bins. Our words are the protein families. And then we have underlying functional modules, which are the topics, that generate our protein families. And the genomes basically are mixtures of different functional topics, functional modules. So this is this plate notation or graphical model for this process. So you have the random variables here, which or each circle is a random variables. The only thing you observe are the words. The plates here, they denote replicate variables, so replicate structures. For instance, this plate here represents the topic distributions. So the distribution, probability distribution over the words, and there's k of them. And then you have the documents, and for each document you have this distribution over the topics that are relevant for it. And each document consists of n words, and then you draw from the topic distributions, you draw a topic assignment for each word, and then you sample from the respective topic the word with a certain probability. And because you use it's a Bayesian approach, so you do this approximate posterior inference, and you have the Dirichlet distribution as a distribution over the multinomials, and these are the parameters for these distributions. And so these are the parameters you need to specify. And we've heard, similar methods or more complicated models yesterday. So we used LDA to infer functional modules in this setting, and we inferred. We used 200 functional modules, so 200 topics that were inferred. And our terms that we use are so-called eggnog terms. They're from the string database, annotation of protein families, and they're annotated across 575 genomes. So we started with an analysis of genomes. This rather complex-looking slide shows you these inferred functional modules. So we use the cutoff basically to define sets of functional modules, or the sets of terms that had the highest probability in a functional module. So our functional modules then were no longer the probability distributions, but just a number of terms with the highest probabilities. And then we compared them or checked them for their support based on known functional interactions. And we used the string database, which is, I think, run by Christian von Mehring, and they collect information on functional couplings or functionally linked protein families from all kinds of sources, and they also tell you what the source is. And they also use co-occurrence information. This is what we removed, because we're using the same source of information. And then we selected high confidence pairwise interactions and checked how strongly our modules are supported by that. And there's different measures here. So this is the black curves are the fractional saturation with pairwise links between protein families in the modules. So if you just consider all pairs. And then if you also consider transitive interactions, these numbers get a bit higher. We did some statistics and see that it was highly significant, this enrichment. So we do find something meaningful. And the names here there's 3 modules here which we give names, and these we could also are very nicely mapped to known KEGG pathways. So example here, you see for the so-called vitamin B12 module, this is the KEGG map of the porphyrin metabolism. And the blue enzymes here is everything that's part of this map, and the red ones are the ones that we identified with the modules. And here's a KEGG map for the flagella apparatus, and so the flagella apparatus is needed for motility. Basically, if a bacteria has a flagellum, it can move and can propel itself towards some food sources. And so for this apparatus, we found in one module all the red marked genes here. Of course, we're not the first to ever discover this, so other people have seen that you can discover this from data sets using different approaches, but it was a nice confirmation that the LDA infers useful things from the data. And then, so then the next step, Sebastian tried to find functional modules that are relevant for a particular phenotype. And he first began with using different extensions of LDA, so there's I think they're semi-supervised approaches, so basically there's methods that use sets of functional modules, and they use the modules as features in a multivariate classifier, and then they jointly optimize over the modules that are inferred and optimize the predictive accuracy with these classification functions. However, that didn't work so well, so what in the end we did was that, so for this problem, what in the end we did was just rank individual modules based on their relevance for a particular phenotype. And so in this somewhat complicating looking slide, this gives an overview of this procedure. So we have a set of genomes and metagenome bins, There's phenotype labels for some of them. For others, we don't have any information yet, whether they could degrade or not. And we annotate all of them. This was about a collection of 3,000 genomes now, so extended, and we could infer from a larger data set. And then with LDA inferred so-called potential functional modules. And then we wanted to assign whether a functional module is relevant for a particular genome or not. And we could use these from LDA, these topic weight distributions that are inferred. So basically, I think that's what's used in this haplotype phasing information also for assignment. But the problem is that this turned out not to be the best predictor because these topic distributions are basically then the relevance of a topic also depends on how large the genome is. So we didn't want that. We just wanted the relevance of a functional module to depend on how many protein families of this particular module were found. And so we used the fraction of module protein families we identify in a particular genome as the measure, which we then found a cutoff for to predict a protein family, the presence of a protein family, the presence of a functional module in a genome. And then, based on the presence and absence of these functional modules, one can make a prediction. One can see how predictive they are for plant biomass degradation if you have this information. And so we repeated this a couple of times and then built because you have this because you don't know that you find a global optimum with LDA. So we created a set of such rankings and then merged them to create so-called consensus modules, which are our predicted plant biomass degradation modules. And as one way to support this, we map the protein family content of each module to the genomes of known degraders to see if the protein family is clustered in there. So this is independent information, which we didn't use in inference. LDA treats the terms as a bag of genes, and so we could use it to see if things make sense. So these are our plant biomass modules, the 5 modules we found that are most strongly associated with the plant biomass degradation phenotype. And you can see here different measures of performance. So in leave one out experiments, we have an overall performance measure, some weighted form of the F measure, and then we have the recall and the precision for each module. So how predictive is it for plant biomass degraders? How precisely does it map to them, and how large is the recall? So what fraction of the known degrader genomes does it occur in? And so this module, which includes enzyme for cellulose and hemicellulose degradation, occurs in almost all of them. And then, interestingly, the different modules seem to focus on the degradation of different components of plant biomass. So we have a module that has mostly content for hemicellulose degradation. It's also very specific to plant biomass degraders, but doesn't occur in all of them. Then we have a module for the degradation of these pectins. That's another one of those heteropolysaccharides. Has a high precision, but doesn't occur in all of them. This is a rather generic module. It includes enzymes for the degradation of all kinds of polysaccharides, so not only lineocellulolytic compounds, which may be why its precision is the lowest of these modules. So it also occurs in some other genomes of non-degraders. And then we have a module which is interesting. It includes the structural components of the cellulosome, so these proteins, these coenzymes and dockerines. This is again has high precision but low recall. This shows you an overview of the relevant Casey families that are placed in these modules, and you see again these GH5 and different other glycocellulases, the components of the cellulosome, and we also there were also I think 21 protein families assigned to these modules which weren't known to be relevant for plant biomass degradation before. So these now could be interesting to look up in further studies. And we wanted to analyze now how do these PDMs, these plant biomass degrading modules, relate to these known paradigms that I showed you in the beginning. And so Sebastian, he really went through the entire literature and looked for evidence of which module can do or which organism can do what, and this is the result. This is a list of known degraders he was using. These are taxonomic affiliation, whether they're aerobic or anaerobic, and here whether they use cellulosomes or free enzymes. And for some of them, this is not known yet. And here you see the occurrences of the plant biomass modules across these genomes. And, well, there's no association with one of these paradigms. That's the result of that. So it seems they focus more on the degradation of different compounds of the linear-cellulose, and not so this aspect of whether there's a cellulosome or not doesn't seem to be reflected in the module content. And then if we compared now the PDM predictions also, so we can predict the presence of PDMs of these modules in genomes or taxonomic bins. And you see here for all the taxa for which we had taxonomic bins, and these were from different microbial communities, including linear-cellulose degraders. And you can see here the taxa which the modules occur in. So Bacteroides had predictions of the PDMs, and this is in line with things that have been found in metagenome studies. So Bacteroidalis were, for instance, found to be relevant in the Tama wallaby gut for degrading plant biomass. And there's some, also Clostridialis makes sense. There are some which are more interesting, for instance, the Prevotella. They were so far not suspected themselves to be degraders, but just to help in this process. And one can look at, well, now it seems maybe they can degrade after all. And also there's some I can't even pronounce this, some archaeal species that were predicted to be degraders or had some of the plant biomass degrading modules. So that's also interesting. We found one publication where there was mentioned that one Halobacter species was able to grow on some cellulosic substrate in some sentence in +1. So we have to see whether this maybe gives some new insights about who can degrade linear cellulose. And these abundances are for the genomes of known degraders. These are the predicted PDM occurrences for microbial isolate genomes, and the circle sizes denote here how many species were predicted to encode this module. You can see that some of them have this sort of agrees also between metagenome bins and the isolate genomes, but that the ability to degrade linear cellulose seems to be more far spread so far across the isolate genomes, which could also be because here the draft genome reconstructions, they're usually fragmentary, so they're not complete. So it can also be that a module is not predicted because just there's a partial taxonomic bin and it's lacking half of the protein families, and therefore prediction isn't made. Okay, and we, as one means of verifying the meaning of these protein families, we map them also to gene clusters in the different genomes. And you see here we found 81 clusters in the known degraded genomes, including, for instance, this cluster, which is a cellulose degradation operon in one of these degraders. And you see here the predictions from one module M1 and M5. The red ones are from M1, the green domains are from M5, or the green families. So they're linked together in this cluster. And we have more predictions in the other genomes. And hopefully at some point this will lead us to really the last step, to discover some new important enzymes. So in conclusion, what we can show is that this plant biomass degradation phenotype can be predicted, and that the metagenome bins did provide additional valuable information for this inference. We're currently evaluating novel candidate genes with our collaborator from Norway, and because so far we haven't learned much about potential new paradigms, for instance this SUS-like paradigm, but mostly, well, because most our data set comes from organisms using this free enzyme strategy or the cellulosome strategy, we're thinking about maybe targetedly sequencing genomes of organisms that are likely to employ the other paradigms. And yeah. So this concludes this research part. This is the lab I run in Düsseldorf, and this is Sebastian, who, well, you already saw a picture of him. These are and Aaron, they're currently here, and then we have other people that are working on binding methods, Johannes and Ivan here. And we have collaborators that were involved in various of this project, mostly now in what I talked about, Phil Pope, and also Mark Morrison. With him, we've been involved also in the study of metagenomes of plant biomass degrading communities, and originally also with Phil Hugenholz and other people from JGI. So that's how we got into this whole direction. And I want to use this opportunity to make some, to advertise or to announce that there are 2 postdoc positions which we're trying to fill. So I will be transiting to the Helmholtz Center for Infection Research, and this is a part of a larger center that was recently founded, the German Center for Infection Research, or DCIF. And this has locations all over Germany. This is where Braunschweig is located. And in Braunschweig, we will be building up a computational biology resource or group for within the DCIF. And so the aim here is to work on translational medicine projects, and there'll be data generated genomic and medical data relating to different infectious diseases, and looking for people who would like to work on this and to develop statistical methods for the explorative analysis of this data, with the aim of going towards translational medicine. So if you know people that are interested and that have a background in bioinformatics, statistics, or related, good programming skills, and an interest in talking in interdisciplinary collaborations, then please feel free to point that out to them. And finally, the last thing I want to do is announce an initiative, the so-called CAMI initiative. And CAMI stands for Critical Assessment of Metagenome Interpretation. And the aim of this is, or the organizers of this are Thomas Ratti from the University of Vienna, myself, and Alex Djerba, who is also sitting here. And so the aim of this initiative is to provide the community an opportunity to perform an assessment of all computational methods for shotgun metagenome analysis. And we thought to initially focus on assembly and binning methods. So I think it's something that would be very useful to the users.
Selection.
Okay.
So maybe that can.
Thanks. We can talk about it.
And also about the LDA. There is also a hierarchical version of LDA. And if you know something about the structure and relationships on the proteins, perhaps you can also incorporate hierarchy there.
Yeah, absolutely, yes. We've been looking into incorporating some more information and trying different LDA models, so absolutely, yes, that makes sense to extend it.
Yeah, very nice. I'm just curious if you just do very naive predictions based on the sequence of those microbes. For example, just look at the distribution of the tamers. Is that possible? I know you convert everything into protein.
You mean you want phenotype-specific tamer predictions? Or?
You want to predict which microbes predict the biodegradation.
Right, and you want to do it based on K-mers, or?
Is that possible?
I'm not sure if it's possible. Maybe. One would have to try. I'm not sure.
But all those protein families is derived from the sequence, right? The original data are the genomic sequence.
Maybe you could. Maybe you could if you find K-mers that are mapped to certain protein families. I mean, for our task, the protein families is what the experimentalists most want to have. It's not just about building the classifier, but yeah.
So this seems to be another instance, very nice instance of where bioinformaticists act as filters, if you will. There's a lot of data out there, and we're trying to distill it into a form where the experimenters can go and actually test these things. Have you considered using microsynteny or clustering on the chromosome as one more instance of filtering it down?
Right. Yeah, that's something I would like to use. Makes sense. Yes.
But you haven't done it yet.
I've been suggesting it to people for some time. So yeah, we want to do it, and so it's in progress. Let's say it like that.
So the topic analysis that you did, could you use that for the situation where people map reads against KEGG pathways, and then they ask themselves, is this pathway present or not present? Because, you know, if you just say, okay, how many reads have I mapped to this pathway, and don't look at what you've mapped to, then, you know, the pathway's got 50 different enzymes, and if only one of them is attracting a million reads, but nothing else, then it's put it another way, if you just map reads against KEGG pathways, and you want to rank them by which ones you think are present and which ones are absent, which ones are actually present in the metagenome. do you think a document, that kind of topic-based analysis, have you thought about that?
Yeah, we started initially with something like this, but so the KEGG pathways don't seem to be that helpful in that way. Maybe it's because they're too broadly defined. We also tried eco-psych, things like that, and maybe the representation is too. I mean, if you look at the vitamin B12 pathway, just look at how large it is. So this integrates many things. So it's, I think, it's
too big in a sense.
yes,
You
and
can't have
yeah,
everything lighting up because
yeah,
it's not all needed.
yeah.
Okay.
You may have already covered this one, Alice, from Volker's question, but the question of experimental validation, I guess that was related to your, if not the question, that was related to it. What aspects of all of this would you be willing to, if you got to the point of experimental validation, what exactly would it be that you would be doing?
So what we'd be doing is we would start with these clusters here. So so we have the mapping onto genomes with degraders that use unknown paradigms, and then we find interesting clusters and the genes in them, and then we would select a couple of them to be expressed and then tested for cellulolytic activities. So that's what we plan to do.
You would take the whole cluster and try to...
Some genes of them probably not. Maybe I don't think the whole cluster, but a selected set of genes from there.
I mean, it occurred to me that in these sort of pathways, you've got a number of players, and perhaps typically they would all need to be there for anything to happen. So,
Yeah.
I mean, I think it makes sense from your perspective that you're looking for these kind of functional modules, and the logical conclusion would be, well, that's the set of things you would need.
Yeah,
So wouldn't you need to put them all together?
Yeah, there's two ways you can do it. I mean, one is to complement basically an existing mutant where just one gene is missing. So then you would, this is not how it's done here, but how it's somehow done when you try to find genes with certain functionalities and pathways. So you knock out a relevant gene and then you stick in some other genes and you see if that works. Then the organism retains the capability to synthesize histidine or something. What they usually do here is that they just test individual enzymes for activities on some substrates. So that's how they test enzyme activities, and then I'm not sure how they would test for carbohydrate binding modules, but that's what we have. Our collaborator, they know about that. So they would test individual enzymes for selected activities. I think you can also screen for a set of activities.
Yeah. So you describe them as being either degraded or non-degraded, but in reality it's a community, I assume, that is degrading the materials rather than any one bacteria on its own. Or you...
I think it's I think individual bacteria are known to be able to degrade us, so they can do it by themselves. In the community, there can be multiple degraders, but there's also some that seem to help. So I'm not enough of an expert to tell you which organism does which contribution, but there are organisms that can do it themselves, and they live in these consortia or in these communities. And
but your original functional annotation is based on being able to degrade on their own.
Yes, on their own. So the isolate genomes were all tested that they can do it on their own.
Yes, at the beginning you said that you need assemblies to do this kind of analysis, and that's pretty clear. But your second to last slide, you then showed what you can see
This one?
in metagenomes versus what you can see in isolates, and there's a big discrepancy. So
that's true.
have you got any? Can you say anything more about this? I mean, how much more do we have to be? How much does assembly have to approve by, or whatever? How much work is it going to take to get all those dots on the right also on the left?
But yeah, there's two things in there. One is the partialness of the metagenome bins, and the second thing is that we might also be seeing a phenomenon here. We're studying it's a smaller set of these are, so what we're studying is the set of metagenomes we have studied that are included here is also much smaller, right?
Okay,
And,
okay, so if you had way more data, way more stuff.
Yeah, so I would imagine that if we have more, I'm not sure if it's really, so there could be two explanations. Either it's something to do with the data, with the fragmented nature of the bins and with the smaller number of metagenomes we have, or it could be that actually these microbial communities, they tend to preferentially use these few phyla. So yeah, needs further exploration.
Okay.
Okay, yeah, I mean, you don't believe all of those organisms are you don't believe that all of those organisms are really doing degrading in the same. It seems to be it's very unspecific, the genome section. I'd say it's worryingly too many for quite a specific thing.
Well, it depends, but if we extrapolate from our cross-validation accuracies, the prediction of these modules, with the exception of M4, they are very specific. I mean, so they tend to make few false positive predictions if we look at our data set of known degraders and non-degraders. Of course, this set of known degraders and non-degraders is somewhat biased towards certain taxa. So I'm not saying the only module that's unspecific for the degraders or is less specific is this one here. But if you look at the others, at least from our cross-validation experiments, they have good precision. It could, of course, be that because our learning data set is not representative for the entire collection, that we do see some differences here, and that could cause some more unspecificities, but may also be correct.
Under these kind of fun experimental conditions where all of these bacteria are capable of degrading.
It could well be, and it's also there are some estimates also that that actually about 25% of all sequenced genomes they searched for that they might actually be degraders. So it could be possible that it's quite a common trait. So
you have gene cluster and also genomes, but I don't quite understand what the meaning of metagenomic bins.
A taxonomic bin is a set of contexts that was assigned to one taxon. So you have the reference taxonomy of organisms. For instance, a taxon is the Methylobacter. And then with the taxonomic binning, you assign a set of contexts to a particular taxa. And that's a taxonomic bin. Well, if I go back, was a bit quick. So these would be taxonomic bins, right? Sets of, and then if you go to low ranks in the taxonomy, then this can be representative for an individual, or any isolate data from a metagenome. Then this gives you some draft genome, of course, with haplotype variations in it and things like that.
And taxonomic bin is the same as metagenomic bin?
Yes. Okay. Yeah. I'm saying taxonomic bin because there's other people that bin by function and they bin this and that, and so yeah, the taxa, bin by taxon.
So you use a contig or use the read?
This contig.
Similarly?
Well, in our approaches, this is all contig based. There are read based. Well, others bin also based on reads, but so our if you use longer sequences, usually the predictions become more accurate. I do think accuracy is important in some way.
You only divide into 5 bins, right? Have you tried to get lower level?
No. This is just an example. We divide into whatever is possible, as low level as possible.
Yeah, because in your example, you only show 5 bins. You know.
Hold on. You mean here?
Yes.
So okay. The bins are basically, this would be a bin, like Candidatus accumulibacter, one bin, one taxonomic bin.
I see.
And then we say these modules, they occur in these bins. The protein families of these 4 modules, for instance, occur in the Prevotella bin, in the Bacteroides bin.
So there are many bins.
Yeah.
Okay, I think we better draw this part of this session to a close. Thank you very much, Alice.